Hello everybody, this is Luis Escobar, Assistant Professor at Virginia Tech. I'm going to provide an introduction to ecological niche modeling. But first, let's learn about what I am referring to when I talk about a model. Not this kind of modeling, another kind of modeling. The modeling that refers to simplifying complex systems. So we are going to use models to represent things. It could be a theoretical model. It could be, in this case, this is a theoretical model. With theory, we are trying to simplify and summarize a complex system or a complex phenomenon. But it could also be an animal model where, for example, we want to study complex diseases like cancer in humans, but we simplify everything to use, for example, a mouse as a model of what could happen in human bodies. This is another model. Here we are simplifying the anatomy of an animal. And let's imagine that I want to simplify the ecological tolerances or the climatic tolerances of this species, a black bear. So we can represent many things. And in this case, I want to represent the suitable conditions for this polar bear. So to do that, first, we need to understand what's behind the climate or the suitable conditions or the suitable habitats. Because maybe my question is, can polar bears live like in these cold conditions? Could they move from the North Pole in the Arctic to the South Pole in the Antarctic if they have food? So besides representing something like I'm showing here, they can also predict something. So models can be descriptive or they can be predictive. So the first step to make a model is to have observations. In this case, I want to identify what are the climatic tolerances of polar bears. So I need the observation of, let's say, temperature. I could have many variables, but in this case, I want to try with temperature. So maybe one observation is not going to allow me to simplify the complexities of temperature in the Arctic. So maybe I need many observations. And after I have many observations and I have an understanding of the maximum, minimum, mean values, then I can end with a model, which in this case is the average, that summarizes many observations to have one representation of the conditions where I can find polar bears. And in this case, this is my model. This is a very basic and simplistic way to think about ecological niche modeling, where we are trying to quantify the tolerances of my species or the environments or climate suitable for my species. And I develop a model based on a series of observations to have like my good representation or estimate of what my species tolerates. We can also apply this to infectious diseases. This is the case of Marburg hemorrhagic fever, which is a filovirus. And what researchers did was to identify areas where cases were being reported. They collected the coordinates of where those cases happened, and then they had a figure like this, where they have temperature and they have precipitation across the continent of Africa, and they were able to summarize all the climates in Africa in terms of temperature, mean annual temperature, and precipitation. So all these points are the climatic conditions of Africa. But these squares here are the areas, the regions with cases of Marburg. So here we have a nested or a niche of the climatic conditions with reports of Marburg. So we can quantify that, we can measure that, and we can identify the borders of that to find the most consistent climates where we see transmission. And with that, we can then map the rest of Africa or see what could happen in areas where we don't have that data. I'm going to show another example. This case is for a vector-borne disease, cutaneous leishmaniasis. And you can see all the places where I have cases of the disease. So I can develop a map that is similar to this one, but this one has longitude and latitude, and this one has temperature and precipitation. So these two are maps where in gray I show the areas of study. Here I'm showing the climates of study. And in red I'm showing where I have cases. Here I have the climates where I have cases. So with this we can develop some models to, for example, try to fill some gaps. You can see that here we don't have data, we don't have observations here, we have some gaps here because we see gray but not red. So we can reconstruct what are the conditions where transmission of cutaneous leishmaniasis is more persistent, and we can also make predictions. Another example, this time for aquatic ecosystem, is a virus. It's a rhabdovirus, the viral hemorrhagic septicemia virus affecting more than 140 species of fish, so many species of fish with fish kills or these epidemics that are very catastrophic and very dramatic in the Great Lakes, for example. So this is the number of species of fish reported. More and more species are being infected across time. And what I want to show you is that we can see the virus circulating in fish in the Americas, in the Pacific coast, in fresh waters, in sea waters in the Atlantic coast, in Europe, in fresh and salt waters, also in some regions of Africa and some regions of Asia. So we can even identify the different lineages or strains or genotypes of this virus, which is the same species, just tiny variations to show that most of the diversity of the virus happens in Europe, and maybe this is the origin of the virus. And all the areas that are more similar genetically are just introductions. So if we see this, we can compare two different lineages that happen in completely different areas, in the Pacific and in the Atlantic. And if we identify those different, genetically different strains, we can see that it doesn't matter that they are genetically different. They happen in the same environmental conditions. So the conditions are consistent even if there are some small genetic variations between populations. So using this theory, the idea of ecological niche modeling, we can identify what are the tolerances, for example, for the reservoir or the host of my parasite, for example, a rodent. Then we can identify the conditions where we see my parasite. It could be a virus. So you can see that the virus maybe is not happening everywhere where my rodent is happening. Maybe it's happening only in specific conditions, in a subset. And then we can identify conditions, environmental conditions, where we see transmission to humans. So again, you can see here that transmission to humans or spillover transmission is not happening everywhere across the range or the conditions where I see the host, only in specific conditions. And this is the basis for the study of infectious diseases. What are we modeling? Are we modeling cases in humans? Are we modeling the pathogen, where we have the pathogen in different host species or in the environment and in humans? Or are we modeling the vector or the host or the reservoir for my pathogen? And this is all for this introduction to ecological niche modeling for infectious diseases. Thank you.